It is my great pleasure to introduce Chen, who is a professor in the Department of Bioinformatics and Computational Biology at the Anderson Cancer Center. He is also the Fellows of the American Institute of Medical and Biological Engineering and of the Asia Pacific Artificial Intelligence Association. He's contributed to many cancer research initiatives, including the Cancer Genome Atlas, the 1,000 Genomes Human Cell Atlas, and Human Tumor Atlas Network. He has a long track record of developing computational methods to analyze and interpret human genetic, functional, and clinical data, resulting in many widely used software, such as BreakDancer, VASCAN, and Monova. Today, he will present his recent work in cellular modeling for cancer dynamics in tumor immune microenvironment. Please join me in welcoming him to the IICD.
Thank you very much. I'm going to stand here so I can say you more clearly. But it has been absolute delightful trip for me to come here to talk to each one of you. We have such compatible interests and programs, and it's as I show you, it's really a great match between me and also your program. So I'm honored to be here and also to talk to Simon, who's been regularly in the domain. So before I get started, I just want to plug in this kind of advertisement. It's a Golden Conference, Single Cell Cancer Biology. And actually, we're organizing in the March next year in California. Actually, a couple of you guys, Laura and Yellow, also organizing this meeting. So including broad scope, cancer biology, immunology, artificial intelligence, technology development, spatial, single cell, everything together. So I think it's a great setting for you to expose to a broader scope of cancer research and to find out how cell dynamics actually play a role in this whole process, which you're welcome to find it out and try to register if this applies to you. So I like to start some of the research history of myself in terms of how I got involved in cell dynamics and cancer evolution per se. So back to 2008, we were one of the organizations who actually generates the so-called first cancer genome using NGS sequencing at WashU, in collaboration with WashU Genome Center and Tim Lei, who was the person who worked on AML. So this was a project that actually started a series of cancer genomics efforts later on, including a lot of a genome sequencing projects and also GCG and ICGC. As you know, this is actually a fairly important project in the sense of we started to make discovery in a very unbiased, data-driven fashion, because nobody has anticipated AML would actually have this mutation in IDH1, which actually altered the metabolic property of those cells at this point. People has been doing more candidate or hypothesis-driven research. They would have not actually anticipated IDH1 or DNMT3A mutation in those conditions. I think this is an example of where data-driven research is extremely important in shaping where the cancer research is going. And myself has been introduced previously by the hosts. At that time, I developed a program called BreakDancer, which actually identified chromosomal breakpoints from next-generation sequencing data. When you apply those in a patient sample, like AML patients, you're able to identify some unusual translocations. For example, this one is called insertional translocation, which involves a segment of chromosome 15 gets popped up and inserted into chromosome 17. And this kind of a new chromosomal architectural change actually were not detected by standard, cytogenetics, because the phage probe were designed to actually capture reciprocal translocation instead of this, insertional translocation. That's why this patient were not diagnosed genomically in terms of her condition, which turns out to be APL, acute promyelocytic leukemia. and which actually causes a clinical diagnostic conundrum in the sense of what kind of therapy we should be given to those patients. If it's actually APL based on DNA diagnosis, involve PML alpha translocation, oncofusion, then this patient should be put on a targeted therapy, actual treatment. However, if it's actually complex cytogenetics AML, then the patients will be put on chemotherapy. So therefore, be able to apply NGS and those data science technique onto this patient data and to identify this genotype, de novo, actually help us confidently identify this patient as APL and put her on the targeted therapy, which basically help those patients get into a remission in response to that therapy. And also because of this discovery, we're able to actually improve the probe design. So we have a test which costs only $200 because of this to apply to additional patient diagnosis and being able to actually improve the genomic diagnosis as a consequence of this unbiased whole genome discovery. I think this is a good example to tell you, unbiased approach plus data science is actually can very much alter the not just research but also patient, clinical practice as well. Okay, my lab now is actually focused on a much more broader scope than just genomics. We're doing a lot of multi-omics, as you know, and we're doing this at the single-cell resolution to understand the dynamics across different stage of the disease under different therapeutic condition. We're not just looking at omics, which we can measure from the data. We're also looking at, for example, other omics, for example, metabolomics. We're able to compute or infer from those omics data. And we're trying to integrate different computational and experimental data, including structural biology and knowledge we curated from literature together to build dynamic models where we can actually simulate the disease and treatment before they are actually being applied on the patients as a way to kind of help advance, therapy and also kind of like therapeutics development and also personalized therapy. I'm going to get into some of those areas in the following slides. So I just like to start my first subject. How do we study cell dynamics via mutations? This is a subject I know many of you here also works on. And a lot of efforts has been inference-based. Basically, you observe very analytic frequency from bulk sequence and DNA sequence at a different stage. And how do you leverage the observation to actually construct the phylogenetics of those tumor and trying to understand how the phylogenetics were shaped by cancer intrinsic as well as extrinsic selective pressure. I think you are actually very much has very deep expertise in this area. And back about 15 years ago, I was involved into more of a single cell based study of this type of project, where individual cells were isolated and also sequenced to obtain their whole genome or whole transcriptome, and trying to construct phylogenetics at single cell resolution in order to understand the intra-tumor heterogeneity within individual patients across different stages and time. So we actually did quite a bit of work trying to develop phylogenetics techniques that are robust to different noise in the data, including missing and also, sequence errors. And we published a bunch of phylogenetics tools in that area, indicating you can do reliable phylogenetic inference based on noisy single cell data, both from DNA, but also increasingly from RNA and mitochondrial genomic sequencing, as you are aware of. Of course, one of the barrier of doing this is oftentimes you want to do multi-omics, but your experiments will require from different experiment and different modality. So therefore, as John Maironi very well summarized in this review paper, one of the challenging tasks is how do you fuse different experimental, single cell experimental data together, right? So you have two fuse data either horizontally, they're acquired from different different samples from same modality, or vertically, which means they are from the same set of cells, but acquired from study by different omics. Or you have to do this in a hybrid way. So how do you actually link those data sets together in both vertical, horizontal, and also vertical way, and across different time and space? So one of the basic mathematical problem is to figure out how to align those data sets of different cells and different features, right? I guess one of the contribution we have been making this is to extend the canonical correlational analysis framework into this so-called bi-order correlational analysis. Basically, you try to actually design this intermediate matrix Z, which has the same row as the first matrix X, but same column as the second matrix Y. And then you apply CCA iteratively among X and Z, and then Y and Z, until you kind of converge in these two, both of this domain. So in the end, you actually achieve alignment both amongst the cells populations, but also being able to link different modality together through the population level alignment. So if you are able to do that well, you will be able to actually link different modality and across experiments for the same underlying cell population and achieve both feature transformation and also cell label transformation.
Sorry,
Yes. Yes, please.
are you, because you're assuming that each cell set is a different independent sample, right? So are you matching randomly, or are you matching when you're trying to align?
I think here we're assuming we're getting data from the same sample, but different cells. So therefore, there's a similar, if not identical, cell population structure within the data itself. So we can achieve the alignment at least like clone level, if not at the single cell level.
But then,
Hmm?
are you trying to find different configurations and then minimize some kind of cost function that
Yeah. Very good. So if you're familiar, if you're familiar with CCA approach, it's a technique aligning two matrices like this, same rows for different columns, for example. So I guess you derive two different projection matrix U and V transformation, transform those two matrices into the same latent space, where they actually have the best alignment in terms of their L2 cost, L2 norm in that latent space. So this is a so-called CCA, a transformation. So we're just generalize this into a more generic setting where both rows and columns are different. So therefore, you could apply the same projection, type of transformation to align this data from same sample but different experiment and different cells at the same time. Does that kind of answer your question?
Yeah, I was just still slightly confused about if you change the row configurations.
Mhm.
So do you do that? Do you do iteratively calculate the alter normalization and see which rows configuration? So if you arrange cells in rows, different configuration of those rows can give you different altitudes, right? And so basically you try to minimize that altitude.
When you say configuration of rows, do you mean different order of the rows?
Different order.
It doesn't matter because we are trying to regularize on the population structure of the data. So it doesn't regularize on individual cell.
Oh, I see. Oh, it's not. Oh, it's
Yeah, so they can come from different rows. Doesn't matter where they are, but at statistical level, if they form same cluster, for example, they come from same clone, that's fine. The methods will automatically group them and also align them across different matrix X and Y. Does that make sense to you? Yeah, okay. So it's really a statistical alignment between the two different data sets. Okay, but I think one of the inherent issue here, many of you also working on this problem, is DNA is kind of a discrete, right? Digital, like point mutation or copy number alteration at the single cell level, they are kind of a digital type of alteration. But for phenotypical level, no matter you're talking about RNA or other modality, they're more or less kind of a continuous, right? There's a kind of plasticity in the system allow you to have the RNA states diverge from stationary states. So therefore, you need a system that can help you link the discrete entity with this continuous observation you have from the data, right? So this is where we think machine learning or deep learning can help you. Basically, you want to capture some, likely epigenetic regulation or any other complex regulation that helps you link the discrete DNA versus continuous RNA. So this is where another scenario where you can actually try to map your DNA data versus your RNA data in single cell resolutions. Similar concept, although increasingly people have been doing a co-assay where you can acquire DNA and RNA from the same cell experimentally and use that information to help you better anchor those two set of cell population. I think this is, several labs has been doing this experimentally, although it's not entirely high throughput. It's kind of a costly at this point. So therefore, I think some statistical methods help you establish that, linkage is still being needed in this space. Just to give you an example, so this is a breast cancer sample. If you sequence DNA and try to do the copy number profiling, you can observe a copy number clonal structure like this, maybe 4 major DNA clones, copy number clones, or 8 minor clones. However, if you do RNA sequence from the same cell population, you're going to get a very cloudy representation of RNA because of the continuation and plasticity in the RNA space, right? So what you want to do is try to align those two different representation together through this kind of alignment and also plus deep learning to capture the DNA RNA regulatory relationship. So if you do this, you try to fashion initially the DNA is going to cluster different latent space, and RNA is going to cluster separate from the DNA. But as you go through iterations, eventually they're going to converge into the same population alignment, where the DNA and RNA are now actually co-localized into different clusters, basically by their underlying clonal identity or structure. Right, just like here, those different colored RNA cells is now going to be distributed into different DNA clones, also in a different coloring system. And this is actually very important to understand the cancer evolution of those data. For example, if we do a DNA phylogenetics tree based on copy number structure itself, you're gonna see this yellow clone is actually earlier than this blue clone, and those are both earlier than green and this dark blue clone. However, if you look at the aligned, this what we call co-embedding, this is including both DNA and RNA together into this space. You actually saw this yellow clone is actually far away from this blue clone and closer to those green clones. So they have different implications in terms of the distance and space. So this is very interesting because this is suggesting, clone 3 and clone 4 are, what I call one small step in DNA, because the DNA change is not huge, but it's a one giant leap in RNA because it causes a lot of change in the RNA. So this allows you to basically measure the RNA phenotypical difference or phenotypical timing relative to the DNA change or DNA timing. So this indicates those clones potentially have some selective advantage over the other clones. So I think this type of co-embedding allows us to very quantitative, precisely measure this type of evolution and their consequence. And then if you try to do some differential gene expression analysis based on this type of representation, you're going to identify much more informative genes. For example, ERBB2 or ERBB4 is actually one of the top hits. It is actually related to the lineage, loss of lineage marker into the luminal ductal cell lineage. And it's indicating there's a potentially going through accelerated differentiation in this particular cell population. I think this demonstrates the power of integrate those data together to perform kind of a cancer evolutionary analysis. So similar type of approach or study can be applied into immune cells, as many of you potentially have been aware of. Particularly, we are interested in studying the T cell response and T cell fate in the context of immunotherapy. T cells, just like cancer cells, they also have their genetic identity, which are mostly being characterized with different T cell receptors in their cell surface, which recognize different cancer cells as a way to have T cells activated and started to kill cancer cells. However, they also have to be differentiated and migrate into cancer and being able to professionally kill cancer in a tumor microenvironment. So therefore, the genetics and their RNA phenotype has very strong interplays. So their genetic identity basically determines whether the T cell is going to have a different RNA journey or RNA trajectory, right? Many of you know. So this is actually very important because we're trying to answer the question of why certain patients responded to the immune checkpoint inhibitor or combination therapy while others don't. Same cancer type, different patients, same therapy, but the response are actually very much different. Is it because there's a loss of target expression in the cancer cells, or is it because the T cells does not have the required T cell receptor? Or maybe it's because of tumor microenvironment is very immunosuppressive. Which one of those factors are at play and how they actually interact together in real patients? So we look at some of the clinical trial data. This is a particular HPV 16 positive cancer, oropharyngeal carcinoma, head and neck cancer. They were put into therapy. First, they were applied to checkpoint combination therapy and anti-CTLA-4 and anti-PD-1 therapy for 2 cycles. In the neoadjuvant setting, to see if their tumor is going to shrink. And indeed, a lot of the patients actually have tremendous shrink of the tumor, which we call major histological responders. But a fraction of patients do not have a significant shrink. They still have residual tumors, which we call non-histological major responders. So we acquire single cell data pre the immunotherapy and also post the immunotherapy at day 30s. And we compared the single cell gene expression data in terms of the different cell population dynamics and associated with the responder status. As you can see, much of those were match the knowledge of immunotherapy because in the responders, the CD8 and CD4 T cells has a very significant increase in the responders compared with the non-responders. And the T regulatory cell ratio actually has a reduction in the responders compared with the non-responders. So this is also match the immunology knowledge. However, we observe a population called effector Treg cell characteristic expressed in FOXP3 as well as CCR8 gene expression levels, compared with other T regulatory cells, which has a very strong specific, population size reduction in the responders compared to non-responders. So this indicating it's very highly important to have the effector Treg cell population reduction in the responders as a potential as a consequence of those checkpoint therapy response. And interesting, if you look at the responder compared with non-responder in terms of their T cell, CD8 T cell transition pre and post therapy, you see two different state transition patterns. So in the non-responder, you see a lot of transition between the resident memory T cells to the effector and effector memory T cells in a clonal type specific fashion. So the red color representing the clonal types are actually identical between the two states and pre-post therapy. So the darker the color means there's more clonal type that is actually enriched in this kind of a transitional process. So this is a very characteristic different from responder and non-responder, indicating the T cells really need to have the TCRs that recognize the tumor antigens in order to achieve this kind of effect transitional states in the responders. So, oh, this version of slides is okay. And we also observed this in the spatial transcriptomic characterization of the same patient data. For example, this is a and 5K slides of the same tissue sample from those patients. We also designed probe to target specific TCRs. We know our HPV epitope specific. As you can see here from some of the data we actually saw in those medium data, this particular CD8 T cell actually express the HPV specific T cell receptors right next to cancer cells, which are pink cell here, indicating potential interaction between T cell and cancer cells in antigen specific fashion. Okay, so we then have this quantitative science hypothesis. We think the T cell ability, namely the ability for T cells to interacting with T cells, is encoded by the TCR sequence and also their structures. And the tumor infiltrating T cell RNA states, they jointly determine the T cell tumor killing capacity and also patient response in the ICI therapeutics. So therefore, we think if we try to integrate those two types of information together through multimodal modeling, we will be able to better identify the HPV specific T cells and also their T cell receptors. Okay, so of course it's difficult to do this in an unsupervised way. So we actually acquired experimental data using some in vitro assays. This is called functional expression of specific T cells developed by Kelly Smith and Johns Hopkins University, where we actually spike 50-mer from HPV genomes and trying to test those 50-mers together with controls and through a tetramer into the patient blood and see which one of those HPV peptides were actually recognized by T cells in the patient blood. And we also ask whether those were found in the tumor tissues, in the tumor infiltrating lymphocytes. And through that process, we're able to significantly shrink a hypothesis space of about 5,000 TCRs, 500 peptide down to around a couple of hundred. And then we further do in vitro cell assays for us to narrow down to specific TCR and peptide combinations, which actually is going to generate an immune interferon gamma response. And we also further testing those in the tumor killing co-assays in the NS device for us to identify the actual tumor killing T cells and their TCRs. So this actually serves as a labeled data sets for us to train some kind of AI-based model. So, and using that data, it's already apparent HPV-specific T cells and also the bystander T cells, this is flu virus specific T cells, they have very different transcriptomic RNA states in the UMAP. So HPV-specific T cells are pretty much in the tissue resident and tissue effector states, and CEFX are in actually different states. So if we can actually label those cells and try to look at their differential expression, it's already very apparent. We can run an RNA expression signature based on just simple labeled RNA expression data sets and acquire gene expression signatures through a simple logistic regression model. And just based on RNA expression signatures itself, it can already help us identify additional tumor reactive T cells, which we were not able to identify through the experimental pipeline in the previous slides, which we actually have done, and it helped us identify additional candidate tumor reactive T cells. And then we apply a machine learning based approach, which help us co-embed the RNA states, RNA expression with the TCR sequence embeddings, which we actually had a vector representation of the TCR sequences, and we try to align those two type of data together through the graph machine learning approach. And the attention matrix through those machine learning approach are able to help us explain which particular genes are actually interacting with which particular residue in the TCR sequence itself. Help us understanding, what important structural information in the TCR is actually interacting with which genes in the RNA, in their RNA experience, RNA journey. So this approach is also very effective. Without actually having too much of a training data, we're actually able to identify the gene expression modules de novo from those data. As you can see here, this is a module indicating antigen presentation involving HLA genes and also CXL13 and CXL6. And this is a module indicating epigenetic modification of those T cells. Those are completely acquired based on the machine learning. But the importance of this is we can make discoveries based on those data now instead of completely rely on hypothesis-driven approach. So we think this has a lot of promise into the discovery and understanding of interaction between the immune and tumor system in those therapeutic settings. Okay. Of course, tumor microenvironment is very complex, has many factors, not just immune recognition, but also it needs to have the right chemistry, particularly metabolic environment of the tumor microenvironment is extremely important. I know many of you are trying to do this research here as well. So one aspect we think is particularly interesting can be potentially helped by data science is to try to model the metabolism in those tumor microenvironment based on the RNA expression data. So we know a lot of omics, including DNA mutations like the IDH1 mutation we found previously through whole genome sequence, is actually very dramatic to alter the metabolic tumor and tumor microenvironment. So there's a potential to do this through the so-called genome scale metabolic models, which are basically a set of a database, that has already been identified and also curated into the public domain. Because we know a lot of chemical process, including, for example, glucose consumption, pyruvates generation, those are actually pretty much conserved across any organism. And those knowledge are actually very well categorized in database like Metabolic Atlas, which for human contain roughly 13,000 reactions and also almost 10,000 metabolites. And their reaction formulas are very much available. So we can use this approach called flux balance analysis for us to try to link the RNA expression profile to infer the metabolic flux. In this context, it's the reaction rate of each of those chemical reactions together, because those chemical reaction has to be fused, has to be fueled by those chemical concentration, like how much RNA and protein that is available in situ, in those tumor microenvironment. So this is potentially can be defined as a constraint of optimization problem, where we have a matrix constitute matrix which lists out the relation between different chemical, each row representing one particular chemical metabolites, and each column representing a particular chemical reactions is listed here, and we're trying to infer the rate of each of those chemical reactions, right? So this whole thing is a kind of a highway traffic, system where the reaction has to move at a particular rate in order to generate enough chemical concentration in order for the next reaction to occur at a particular rate. So this whole thing is very well regulated at biochemical level, particularly when the tissue or when the organism in a steady almost homeostasis states. So they have to satisfy those kind of constraints. So we can constrain those by gene expression, because gene expression, at a broad scale, constrain how much of enzymes available in the cells which are required to catalyze those chemical reactions. Okay, so this can be defined as a constraint optimization for us to solve the reactions through by solving those differential equation models. And we develop a program called MetaFlux, which allow us to transfer the bulk RNA expression profile from tissue into a metabolic flux profile of each of those tissue samples, right? And because you're a quantitative science program, I'm trying to lay out the mathematical equations a little bit in a little more details. Yes, please.
So these are the reaction rates, right?
So,
V1 to V7 are the reaction rates.
yes, we're trying to solve the reaction rates.
So how can they be negative?
It should not be negative, actually.
But the bounds were some of the bounds of the next one.
This one.
Yes. Yes.
This is not the actual model. This is for the theory.
But the bounds are from negative.
Oh, yeah. This is not what we actually implemented in our program.
Oh, I see.
So our program actually, the bound is actually here. So it's definitely the lower bound is going to be 0 for irreversible reaction. And definitely we have considered those, right? As you know, we have to take into consideration relationship between gene and proteins, because it could be multiple genes involved in generating a protein. It's called gene-protein rules, as you know. And we have to consider the medium condition, what nutrients are available in the media, right? So, and those are all kind of become the lower and upper bounds. And depending on if the reaction is reversible or not reversible, you have different bounds, that kind of thing. And in the end, we're defining this as a quadratic optimization problem. We're trying to maximize biomass or minimize the negative biomass of the cancer, because we're expecting the cancer is going to keep growing, while we also minimize the flux, some of this mass. So this is under those constraints, we have defined. So there are stoichiometric relations, right? I think apparently this, so I'm kind of a little bit skip forward. So that's what we do for bulk. And for the single cell pipeline, we're doing this at the cell cluster level. So now each cell cluster can potentially has an independent stoichiometrics and independent reaction rate, because they are likely different cell type. Tumor cells, immune cells, they can have different biochemistry, different reaction rate. So we have separate matrix for each of those. We're trying to have a matrix set up, there's communication between those different cell types through the tumor microenvironment. They still have to follow the same biochemistry constraints, but they will actually allow them to communicate. So we'll also have a matrix P to allow us to take into consideration different proportion of cells. There's more tumor cells, less tumor cells in different tissue. So therefore, those all being incorporated into the scoring function to influence the reaction rate inference in the process. Okay, so of course, we validate those into cell line data first. This is from IICD cell line, where we have the true metabolic assays at the metabolic flux, from experiments, and we compare our computational prediction with those experimental, observed flux, and the correlation coefficients about 0.8 for most of them. In terms of the directionality, in terms of consumption or segregation, for example, because you have two directions for each metabolite reaction, the correlation is also pretty high compared with some other algorithms. And so that's on the cell lines. And we also look at into the tissue. I compare our inference, for example, this is for glucose, I think, consumption versus the, PET, FDG PET, which are the experimental way to, through imaging, to look at the flux, measure the flux. So the correlation coefficients also reasonable, 0.3. And we also trying to look if our inference is indeed, makes sense when we're trying to randomize the medium condition, because again, we want this to be biologically meaningful. When there's no glucose in the media, for example, we're not supposed our model should be making useful inference. So therefore, we randomize what's available in the media or not to generate a null distribution, and then our model actually is producing statistically significant results compared with those random background, also indicating we're actually performing biologically meaningful inference when our computational condition matches on the actual medium condition where the data were derived from. So we are actually confident our model is actually generating biologically meaningful results. Okay, so this was further examined into in vitro data where we're trying to co-culture cancer cells. This is a B-cell lymphoma with CAR-NK cells into NST mice across, 5 weeks, about 5 weeks time. And we have different experiments. This is just tumor. This is tumor plus wild type non-transduced NK cell, and with CAR-19 NK cell, CAR-19 and IL-15 NK cells. There are different type of NK cells being co-cultured with cancer cell across a time. And we have the single cell data from each of those conditions, and we can see the different cell state transitions. This is a tumor, this is NK cell, this is pre-therapy, this post-therapy. Once the cells encounter tumor cells, that started to transition into different states. And those are single cell dynamics. As you can see, different product, IL-15, CAR-19 product is able to constrain tumor cells for much longer, up to day 14, before the tumor started to come back. However, in other type of NK cells, they fail to contain tumor, very early on. So this is a exact cell dynamics characterization we're able to obtain from the single cell RNA sequencing, also represented here, for the same message I just delivered. Okay, so we apply our algorithm on that single cell data sets. For example, now at day 7, we're able to compare the metabolic, capacity of those NK cells. This is 3 type of NK cells: CAR-19, IL-15, and non-transduced NK cells. And we're looking at the O2 consumption rate of those cells. This is computational results for the day 7. As we can see, IL-15 cells has more proficiency in terms of O2 consumption and also proton release, release low protons. And this matches very well with the CF-WORS, which is actually experimentally on the same data. I use the trains map pretty well. So if we're trying to derive a computation score, in terms of tumor flux versus NK cell flux, so this is a relative ratio of those. And in terms of oxygen computation, we can say this group of NK cells actually is the most proficient in terms of O2 computation against tumor cells, because they have tumor flux much lower than NK flux. Therefore, the ratio is being lower than the 2 other NK cell product. And if we use the same metric across temporal time domain, we can see for this particular NK cell product, the tumor eventually actually wins. The tumor computation, initially we were able to, the NK cells are able to consume more oxygen, but eventually the tumor cells are able to consume more oxygen than NK cells over time. And same for the glucose. So therefore, the metabolic persistence of those NK cells actually reduce, relatively speaking, to the tumor cell. Also, in terms of the pH level lactase production, again, over time, the tumor microenvironment become increasingly acidic, and there's more lactase being released into the tumor microenvironment by the tumor cells. So those are all computationally predicted results, which basically it is generating some hypothesis for additional experimental to validate those. Okay, so given all those, how am I doing in terms of timing? Okay, I'm trying to finish up in 5 minutes.
So is it that the tumor cells become more efficient over time, or is it because there are more tumor cells outcompeting NK cells?
The population size is normalized, so we think it's individual cells being lost that advantage. I mean, NK cells over time, maybe because they are, they are enter different states, they're more exhausted, that sort of thing. Tumor cell definitely has heterogeneity because we have different tumor cell population coming back. Initially, the tumor cell population being contained are those guys. But over time, a different subclone actually coming back, which are presumably more metabolically competent tumor cells. So of course, we have deeper analysis into their gene expression profiles and try to understand what are the molecular processes involved to produce that resistance, metabolic resistance signature. So yeah, for sure. I think there's a lot analysis. This is actually published in our Science Advanced study. You can potentially look into some of the details in those papers. Okay. So I know my talk is a little maybe a higher level because I'm more in applied space than in the deep data science space nowadays. So I'm trying to kind of balance those two domain. So but I think the last subject I'm trying to cover is really trying to model cell dynamics, right? I think this is where you guys are expert and can make a lot of contribution. We're still actually trying to explore this to some degree. I think the central goal is similar. We want to simulate the course of cell therapy, right? Because increasingly we're talking about how to transfuse different cells into patients, and what type of a cell we should transfuse into patients, and at what type of ET ratio, right, effector ratio. Are we doing combination or not? And how do we apply this this treatment? Are we doing this multiple dose, and what are the dosage level we should be applying? So those are all very much clinically relevant questions. And I think it's useful to do some kind of a computer modeling or simulation to answer those questions, both for us to develop better experimental therapeutics, but also actually at some point to advise patient therapeutics for clinical decision making, right? So we're trying to at least explore the subject, trying to simulate the course of cell therapy in clinical. And given the tumor and TME profiles, we're able to measure from patients, either at bulk level or maybe single cell or maybe even spatial transcriptomics from those patients. And are we able to incorporate those real patient data into our model so the model become patient specific or individualized? So that's the kind of a goal for doing this type of modeling. Of course, I'm coming from the training of engineering physics. I'm very much, in this realm. I think it's important for us to actually build those systems in order to actually know we have an understanding, right? Right now, I think we're in a realm of we're observing a lot of important things, but we actually don't have the guiding principle rules for us to translate those into practice. I think it's still in a relatively early phase of understanding the theory. So for me, I think my goal is to at least trying to be able to make good predictions, because that's an indicator of whether we actually understand the mechanism of those, therapeutics or those biology. So I think this is a very promising or very important area to continue research on. Of course, as I mentioned, if we can build those models, we can at least help reduce animal testing and amend experimental testing of those systems, and we can help accelerate or examine the particular therapeutic design, and we can also help realize personalized therapy. Okay, we want to do this in the cell therapy context where it's very active in MD Anderson and also have labs who's doing the mouse experiments where we can obtain, relatively more data from. Of course, this is going to be a very challenging project. As you know, there's so many factors affecting the cell therapy response, right? Both in terms of the, different type of cell, they have different biology, and our data actually is snapshots, right? We're not actually getting continuous measurements. How do we construct the continuous dynamics? Of course, the biggest issue is heterogeneity. The cells are different. Each one actually not just different from clone level, but individual cells are also different. And the patients and also tumor microenvironment are also very different case by case. So, but since we now have increasingly more single cell spatial data, we think there's an opportunity to actually transfer those observations into more of a cell level modeling, right? Not just model clone, but actually model cells and cells, how this play out. So there has been previous work, right? This is not entirely science fiction. Previous work, for example, this paper in Nature Biotech has already demonstrated you can model this using an ODG model. This is actually real patient data from B cell, CAR T cell therapy, right? Those are actually clinical data where 3 different response group, complete responder, partial response, non-responders, they actually have different cell dynamics in terms of the CAR T cell proportion over clinical course of time. Of course, the tumor cells are going to have different dynamics. Responders almost has the tumor cell burden significantly reduced and also not increasing, rapidly. But non-responders actually pretty much has a steady tumor burden as an indication of non-response. And a simple model like this, where you're trying to model multiple T cell states, memory T cell, multiple effector T cell states, and T cell exhaustion states, and also a tumor cell and tumor antigen presenting tumor. This is a B cell tumor, therefore it says B. A simple, dynamic system like this, hashed out by ODE, can already pretty much fit in those clinical patient data very well and help us understanding, those different different patient groups actually have different parameter space, right, based on the parameter fitting. And there's different importance of different parameters. For example, the tumor, this I don't remember TK50. This is potentially the TI50, TI60, the tumor proliferation rate, and there's different interaction strengths between tumor cells and also growth and death parameter of those immune cells are actually the most important driver of this whole dynamics, right? You can already being able to obtain those information from ODE model. This is not at cell level, this is a whole sample patient level modeling, right? So we're trying to do this more into cell level now, which is we've started to explore more agent-based modeling. I think some of you even here has been trying this before, as I find out very pleasant way in the early morning meeting with you. So agent-based modeling is a approach where you can actually define a set of agents, and each agent has a different set of parameters representing their cell intrinsic properties. For example, for T cells, you can model T cell proliferation rate, you can have T cell death rate, you can have cytotoxic range, and you can have exhaustion range. You can define the transition rate between those different states, right? You can have many of those parameters, and each cell actually can have different values, right? In this case. And basically, you can simulate hundreds, thousands of those cells and let them actually play out into kind of a simulation or process. This is like a game, right? If you play those games before, Age of Empires, if you even Minecraft, we're exactly doing the same thing, although we're using biologically informed parameter settings for us to actually developing those systems. So this is the system we built for CAR-NK therapy. So we define multiple NK cell states, cytotoxic NK cell states, exhausted, and also vigilant. With this, the memory NK cell states, we have tumor cell states. Trying to obtain parameters based on both literature as well as single cell RNA sequencing data we obtain from those mouse and patients for us to initiate those model parameters. And then we try to have those cells to play out into the computer simulation system, and then we started to observe their dynamics over time, right? So we can calibrate those systems using in vitro assays, including cell line co-culture experiment I showed you in the previous slides. And through the parameter fitting, we're able to actually learn what parameters actually best explain the fitting of the experiment to the in vitro data. For example, we observed, proliferation, serial killing capacity is a parameter in your model, and also the ET ratio. Those are actually the most important parameters for the model to explain those experiments. And once you have those, you're able to make predictions, right? You can use part of the data to do the training and then predict into future, because you obtain data from limited duration. What about the longitudinal, fate, future of those experiments? We can make predictions now, and we can also ask different questions. For example, do we apply different dosage, and how are the dosage scheduled? Is a fractionated, or maybe we just apply one single dosage at the beginning to one of those and be optimal for experimental conditions. So we can actually use a system to generate those predictions. Okay, so this is how it works. Basically, that was the experiment I explained before. This is a parameter fitting. So those dots are real experimental value, and those straight line and also the bands are model predictions. As you can see, for different experiments, we're able to reasonably fit the experimental well, and we're able to, using our model parameters to explain what actually happened. For example, if you look at the different NK cell states, you'll see, cytotoxic NK cells actually in the good NK cell therapy, IL-15 is able to maintain a large cytotoxic NK cell population over a long period of time compared with the two other products. In terms of exhaustion, this is like same guy grains actually has much less exhaustive NK cell states over time than other two type of comparators. In terms of memory, interestingly, this cytotoxic NK cell started to develop memory after 11 days, while the other two type of NK cell never developed any memory, in the process. So this is what our model is trying to help us understand what is happening in the experiment. Of course, those are hypotheses, but at least it's a knowledge-informed hypothesis, not based on some kind of random, guess. This is what the model based on knowledge is actually telling us what's actually happening in those data. And in terms of tumor, you can see, tumor can die many different ways. This is actually apoptosis. This is actually immune cell killing of those tumors. They also demonstrate different population dynamics across different therapeutics contexts. So this actually, again, generated a lot of insight in terms of what the underlying cellular dynamics. Importantly, we're able to outperform ordinary differential equation model, which I introduced to you 5 slides before, which are actually sample level model, right? We're trying to recapitulate, re-implement this nature biotech model on the same data, and we compare our cell-based model against sample-based model. As you can see, in terms of model fitting error rate, in terms of different metrics, we're able to actually significantly outperforming sample-based modeling, representing indicating the importance of model cell level, stochastic or heterogeneous information into this type of settings. So this indicating, our efforts is actually making a useful contribution compared with the traditional approach. Okay, that's a quick conclusion. We think cell level modeling, either by agent-based or some other approach, is going to be really help on CAR-NK or CAR-T type of a model. And the important parameters we learn are the effector to target ratios, cell filling capacity, and also proliferation rate of those NK cells. So therefore, if you're doing cell therapy development, those are the important aspects potentially you want the cell engineering to optimize against. And of course, we're trying to model more complicated system to include, for example, metabolism, to include other aspects, TME features into the model. Right now, it's largely single cell based setting instead of spatial. We want to expand this into spatial setting. Those are the kind of future perspective. So this is a computer simulation we have, now, exactly as promised, we can simulate the tumor cells from time 0 to, day, pseudo time, day 14, 13, and this is a NK cell initially at the boundary and now infiltrate into the tumor and kill the tumor. So those are the population dynamics generated in silico. As you can see, the dynamics is actually pretty complicated. It's not a simple, curves or something, because this is a lot of interaction among those cells in a kind of knowledge-informed fashion. So we think this potentially carries some value into the understanding of the biology or maybe the therapy development. So with that said, I'd just like to acknowledge my research lab. They are very diverse, coming from many different, area, not just computational, but also cancer biology, immunology, and we do a limited amount of AI to some degree, but I think we're mostly focused on statistical and data-driven, discovery. And also acknowledge a lot of collaborator and also funding agency, NIH, separated, and also Chen initiative to funding our research. With that being said, I'm happy to take any questions. Thank you for your attention. Yes.
Go back to the slides where you have a small difference in DNA, but a huge difference in RNA. This in quite obvious. So I'm wondering if the distance in the latent space has any meaning, because usually it is a projection, right? So just by looking at distance in the latent space does not inform you about the distance. Right?
Yeah, I think it's difficult to say there's an absolute meaning of this. I think everything here is relative. There's two relative aspects of this. First, this tree was constructed based on copy number difference. We don't actually know the rate of copy number evolution. As you know, it's non-linear because you can all of a sudden acquire a lot of copy number operation. Unlike SNPs, they are relatively fixed, rate. So therefore, the distance on this tree itself is already a little arbitrary or relative, depending on the metric you use to construct this tree. So DNA itself is already relative. And this distance here is the integration of DNA and RNA. So it's RNA on top of DNA. So even though we're kind of using DNA as our basis here in this comparison, so relative to DNA, RNA is actually has a bigger difference relative to that copy number duration, right? This is indicating some kind of a double hits, right? It's kind of the cells trying to not only acquire genomic alteration, but they're trying to acquire particular genomic alterations which allow the RNA to be even more different than otherwise. So it's more of a under selection, right, of those cells. So I guess this is to some degree only tell us the relative advantage or difference of those clones relative to one another, rather than having an absolute interpretation. But I think your point is good. So if we try to maybe include SNP or whatever into this tree construction to construct the tree, and therefore have a relatively absolute timing in terms of how many days or how many years those DNA clone has evolved. And we're trying to somehow be able to measure the RNA distance, which I think is difficult because we don't know what transcription factor we need to modify or what promoter region we have to methylate in order to generate X amount of RNA modification. So that's actually a little difficult, but at least in doing that, we're able to produce more absolute measurements. Hopefully one day to relate this amount of difference back to the actual time, of how old those clones are, right? I think that's actually a good point. Thank you for the question.
We have to wrap up, so let's thank, yeah.
Thank you very much.